We are pleased to welcome our speaker today, Mélanie Brunet. Mélanie recently defended her PhD thesis at the Chemical and Biomolecular Engineering Division at the University of Illinois Urbana-Champaign under the direction of Mary Kraft. Today, Mélanie will talk about three dimensions intracellular biomolecules distribution with a novel technique that she developed on the NanoSIMS instrument. So, Mélanie, please go ahead.
Thank you so much for the kind introduction. Let me share my screen. Wait, okay, so let me do the laser pointer. There you go. So thank you so much for the kind introduction. So yes, my name is Mélanie Brunet. I recently finished my PhD under the guidance of Professor Mary Kraft at the University of Illinois Urbana-Champaign. And today I'm very excited to be sharing a portion of what I did in my PhD work regarding nanosims imaging. So my work is entitled Depth Correction of 3D Nanosims Images Reveals the Intracellular Distributions of Biomolecules in Cells. So to understand cellular function and dysfunction, it is crucial to visualize biomolecular distribution inside cells. So for example, visualizing the distributions of lipids, cholesterol, and other biomolecules, it's crucial because deviations from normal patterns correlate with changes in metabolism and also homeostasis. Additionally, visualizing the distributions of lipids and cholesterol and disease cells also offers valuable insight into the molecular mechanisms underlying these diseases. So for example, in SARS-CoV-2 infection, alterations in lipid metabolism occur, and this leads to increased lipogenesis, and it also leads to alterations in the transport of cholesterol. Therefore, visualizing the distribution of biomolecules or specific lipid species in cells provides information about cellular function, dysfunction, metabolism, and may also yield insight into their functional roles in both healthy and diseased cells. So the distribution of various biomolecules within cells has been investigated using secondary ion mass spectrometry or nanosims. So nanosims maps the elemental and isotopic composition at the sample surface by collecting up to 7 different mass to charge ratios in parallel with a resolution as good as 50 nanometer lateral resolution. And I do want to say that for those of you who are probably more familiar with, say, TOF-SIMS, nanosims does not have a separate spotter step. So go to the next slide. Yeah, so to permit detection with nanosims, rare stable isotopes are selectively incorporated into the molecules of interest. Now, the reason why we need to do metabolic labeling is that if you don't selectively incorporate these rare stable isotopes, you will not be able to distinguish the molecule that you want to image to other molecules that are native in the cell. So during nanosims analysis, a cesium primary ion beam sputters a sample of the surface, generating monatomic and diatomic secondary ions by chemically analyzing approximately the top 5 nanometer of the sample. So the counts of the secondary ions detected at each pixel are used to create a chemical map of the elemental or isotopic composition at the sample surface. And I do want to say, in other words, this technique, what it does is that it creates a chemical map of the species of interest in your sample. So we don't just image the surface of the cell, we can also image inside the cell. So we can do this by using depth profiling. So during depth profiling analysis, you can either image a portion of the cell or even the whole cell, and then you image it many, many times. So then what you get is a series of 2-dimensional images of the region that was analyzed. And then we can also, by doing this, we can also create 3D images. But before I proceed, I do want to say, to improve understanding of how this technique works. So nanosims, when it images the surface of the cell, it removes the outer layer of the surface, kind of like peeling an onion. It does not chop the sample flat. So this, what I'm saying, is going to be useful for the next couple of slides. So as I said, we can create 3D images of these using this stack of two-dimensional images. So for example, if I want to create 3D images of cholesterol, what I would need to do is that I would then just need to stack the images in the order that they were acquired, resulting in a 3D image, say like this one. Now, unfortunately, when we do this, it creates a distorted 3D image. Let me just explain why this image is distorted. So this is the region that we analyzed. This is the secondary electron image of that region that was analyzed. So if we look at this portion of the cell here, we see height differences between this point and this point. This is taller, this is lower. Now, if we look at those same 2 points in the 3D reconstruction, it's here and here. We see that this image is completely flat and those 2 points are on the same height. So that's incorrect. Another thing is that the edge of the cell, it's placed at the lowest portion of the cell, whereas on the reconstruction, we see this edge here is placed at the tallest portion of the reconstruction. So that also tells me that this is wrong. So unfortunately, these discrepancies result in distortions only in the Z direction, not in X and Y, just in Z. So a natural question that arises is, how can we depth correct these distorted 3D nanosims images in order to generate more interpretable 3D images? Well, to address this, we took on the challenge to develop a depth correction tool. So we envisioned this tool being characterized by the following key qualities. So first, we wanted our depth correction tool to be standalone, meaning that it only requires nanosims data. That's it, no other correlated methods, just nanosims data. We want it to be backward compatible, meaning that it's compatible with previously acquired nanosims data. So meaning that if you took data 10 years ago and you want to depth correct it, you should be able to use our tool to apply depth correction. And also we wanted it to be time efficient, meaning that it's fast to implement. So with this list in hand, the next question that we faced was, how do we even begin to develop such a depth correction tool? So as we proceeded to answer this question, the first obstacle that we faced was acquiring sample height. But then came the question, how do we acquire sample height without relying on correlated height measurements such as atomic force microscopy? Well, to answer this question, we hypothesized that we could extract height information directly from the secondary electron pixel intensities to create the cell morphology. And then we would use the cell morphology to shift the voxels in the uncorrected 3D image according to the heights encoded in this morphology. Now, how exactly do we make these morphologies from secondary electron images? Well, first, what we do is that we extract the texture directly from the pixel intensities in the secondary electron images, which looks something like this. And then for the main cell height, what we do is that we create what we call a step pyramid. So the remainder of the image planes, we convert them into binary images, meaning 0 and 1. And then we add the thickness of these binary images, resulting in a step pyramid like this one. And to achieve our final morphology reconstruction, we just add these two components, resulting in a morphology like this one. Now, this is a morphology only for plane 1. For this data set, we had 600 images. So we need 600 morphologies for each image. We only have plane 1 here. So the natural question is, well, then how do you do the other morphologies? Well, that's what I'm going to answer now. So for that, we had to assume that this was our first step at doing this. So we assumed a uniform material removal. With this, I mean that on each location in the sample, the same amount of material was removed. So taking this into account, this assumption, we had previously calculated the sputter depth per plane, which is the amount of material that's removed on each plane, and to, let's say, reconstruct the morphology for plane 300. What this code does is that it subtracts the sputter depth per plane for 299 times, and then we get this morphology. But say, what about for plane 600? Okay, well, for plane 600, this tool subtracts the sputter depth per plane 599 times. So basically, we have 600 images. We will have 600 morphologies, basically a morphology for each image plane. Okay, so now that we have our morphologies, we need to assess whether they are correct or whether they're not correct. So that's the next step that we did. So we performed this by comparing basically the surface features on our morphology reconstructions to the surface features seen in the secondary electron images. Okay, so let's just quickly do this comparison. Let's start with plane 1. So on plane 1, we see that this cell has a couple of ridges, we can see here. And we also see that our morphology reconstruction does capture those ridges, as well as the basic shape of the cell. We were able to capture it. So this good agreement suggests that this strategy produces an accurate morphology reconstruction for plane 1. However, we did notice that beginning from plane 200, we started noticing some discrepancies, and these discrepancies got even more evident by plane 600. So let me just give an example. So for example, on plane 600, we see this circular feature here. And when we look at our morphology reconstruction, we don't see that circular feature. We just see a couple of ridges. So, you know, because of these inaccuracies, then we concluded that, yeah, although morphology reconstructions were accurate for plane 1, they were not so accurate for deeper image planes. So this indicated that our assumption of a constant uniform material removal was incorrect. Now, I do want to say that despite these limitations, this reconstruction strategy still was able to produce morphology reconstructions just using the secondary electron images, which, you know, was still better over the uncorrected images. Nonetheless, recognizing the need for improvement, our next goal was to improve this reconstruction strategy so that we could accurately capture the surface feature, the changes in the surface features that occur due to non-uniform material removal. Okay, so we did have to improve our reconstruction. And I do want to say, so unlike the previous one where I explained that we build the morphology for plane 1, and then from there we subtract, we don't do that anymore. We still maintain the height and the texture, but then the process is going to be slightly different. So for this process, we first start by generating a contoured base, and this is for the main height. We achieve this by averaging the pixel intensities in the secondary electron images just for the very last plane. Now, to get step pyramids for the remaining image planes, what we do is that we convert the remaining image planes to binary images, and then we add the thickness of these binary images into the contoured base, resulting in step pyramids for each of the corresponding image planes. So now we have the main height. How about texture? Well, texture is the same. We derive it directly from the secondary electron images from the pixel intensities. And then to get the final morphology reconstruction, what we do is that we add the texture and the step pyramid, resulting in an independent morphology for each image plane. Okay, now let's just quickly compare and assess how well these reconstructions are matching the secondary electron images. So for plane 1, again, we do observe same ridges, the basic shape of the cell is captured, so that is perfect. We also saw that with our previous method. But what about for deeper image planes? Well, let's see. I did mention this circular feature. And if you can see, now the morphology reconstruction is capturing this circular feature. And if you don't remember what it looked like before, this is how it looked like before. We had some ridges, and now we have a circular feature. So basically, this assessment told us that, okay, our morphology reconstructions are accurately capturing the smaller surface features that are in the cell, meaning that our reconstructions are accurate. Now, we wanted to take this assessment one step further, and we wanted to do a more quantitative validation. So for this, we did correlated studies with atomic force microscopy, or AFM, which for those of you who are not familiar, AFM, what it does is that it records the topography or the height of the sample. Now, when we were working on this project, Unfortunately, it was beginning of the pandemic. The nanosims that we typically use is located at Lawrence Livermore National Lab, so we didn't have access to the instrument. So then we have to find an instrument on campus that would simulate the depth profiling process that occurs during nanosims. So for this, we used FIB-SEM. So FIB-SEM uses a gallium ion beam, and it produces secondary electron images. So what we did is that we took AFM of various cells, and then we performed FIB-SEM. We collected secondary electron images. And first step, we reconstructed our morphologies, and then we compared to see if the texture, if the surface texture was captured. And you can see that the cells are a little bit spongy with deeper image planes, and we can see that spongy appearance in our morphology reconstructions. Also, the shape of the cell, again, is captured well. So that's perfect. Now, as I said, we wanted to do a more quantitative validation. So for this validation, what we did is that we calculated the normalized height difference. So to calculate the normalized height difference, what we do is that we have the AFM height data, we have our morphologies, we normalize both data sets to their maximum, and then we subtract their heights. So values that are closer to 1 mean that this normalized height difference, or basically there's a high error, meaning that the shapes do not match. And values that are closer to 0 mean that their shapes match, or basically lower error. So we started off by comparing the AFM data with the uncorrected morphology. So uncorrected morphologies, it's just what results from stacking the 2D images, one on top of the other, which results like literally in a block. So this comparison is shown on this figure. And we can see that there are high errors on the cell's edge and on the cell's lower portions, which was expected. We did the same thing comparing the AFM with our morphology reconstructions, and we can see that now the errors are pretty low, so they're more towards this region. There are some regions that have a little bit of a high error, which are some edges and some ridges here, but overall, this analysis shows that our reconstructed morphologies have an average accuracy of about 90% compared to AFM, which basically validates the use of pixel intensities in secondary electron images. Now, this is very important because depending on how accurate these morphologies are, it will depend on how accurate our depth-corrected images are, right? So we want our morphologies to be as accurate as possible so that our depth-corrected images are as accurate as possible. So before I proceed to explaining how depth correction works, I do want to remind you again why we needed to do these morphology reconstructions. So we have our morphology reconstructions that we extracted directly from the secondary electron images. And remember, the heights encoded in these morphology reconstructions will be used to shift the voxels in the uncorrected image in order to depth correct these 3D nanosims images. But then how do we perform depth correction? Well, depth correction works the following way. I will simplify my explanation. I'll just take a cross-section here in the morphology. I'll just use one image plane. The cross-section, you can see it here. And I'll do the same thing for the uncorrected enrichment image. So here we have only one image plane of this is cholesterol. And if I take a cross-section in the same region, we see that the voxels are completely horizontal because this image is completely flat. So when we depth correct, what we do is that this tool shifts the voxels according to the height that's encoded in the morphology reconstruction. And it results in a depth corrected image like this one. Now, this is just for one image plane. When we do it for all of the image planes, then we go from uncorrected, which is shown here at the top, to depth corrected, which is shown here on the bottom. So let's just quickly compare how much depth correction improves visualization of the intracellular features. So we see these cholesterol-rich tubules. They appear to be tubes on the uncorrected image. And then when we depth correct the images, now these, what were tubes, are now more like spheres. Also, on the sphingolipids, we see them dispersed with like no clear arrangement. They're just scattered throughout. And after we depth correct, we then see that some of these sphingolipid-rich features form spheres that are near the cholesterol-rich regions. And now the question you may have is, well, what else can we see inside the cell? Because this looks pretty interesting, but we can even see more. So with our developed codes, we can also take cross sections, and we can even take a peek even further inside the cell. So what I'm showing here on the right are the cross sections. Here is a top view of our morphology, of the depth corrected images. And this white line illustrates where the cross section is taken at. So this first cross section is for sphingolipids, and we observe sphingolipid-rich feature here that's about 500 nanometer. And then we observe these two sphingolipid-rich rings. They appear to be empty, at least on the sphingolipid image. But now when we go to the cholesterol image, we see around the same region, we see these, on the same region, we see these two cholesterol-rich globules that are about 1 micrometer in size. And then even more interesting, when we overlay both of these images, we see that these cholesterol-rich rings are encasing the cholesterol-rich globules, and they're not just encasing them, but then on the periphery, they're also co-localizing. So based on shape and composition, we believe that these features may correspond or may be late endosomes, which are sorting stages for membrane molecules. However, it is important to know that the primary goal of this work was not organelle identification, but rather the improved visualization of these intracellular 3D features. So this tool can most definitely be complemented with organelle labeling in order for organelle identification and also improved visualization of features in 3D. Okay, so up to this point, we've developed our depth correction tool. At this point, we wanted to extend its applicability to other data sets. So to achieve this, we obtained data sets from different users. So for example, we had data sets from Christelle Guillermier, which is the director of the Center of Nanoimaging at Harvard. We also got other data sets from the Kraft Lab, and also other data sets from Peter Weber, which is the staff scientist in charge of the nanosims at Lawrence Livermore National Lab. So we began exploring diverse areas, such as evaluating or investigating the 3D distributions of RNA and DNA within cells, also investigating the spatial relationship between influenza virus, sphingolipids, and cholesterol, and visualizing the incorporation of 15N and 13C labels inside marine algae. So we will explore each of these applications in more detail, but then we will start by visualizing the spatial distribution of RNA and DNA. So as I said, these data sets were provided by Christelle. So her aim was to map the 3D distribution of RNA and DNA trafficking. And she also wanted to investigate if there were any potential differences in the distribution of RNA and DNA, both within or between young cells and aged cells. So while working with Christelle's data sets, we introduced a new capability to our toolbox, which is 3D region of interest isolation. So using this new capability, we were able to better visualize the spatial distribution of RNA and DNA in these 3D depth-corrected nanosims images, revealing the following insights. So for RNA, for example, we see that it clusters in these 3 smaller spherical regions and these 2 larger ovoid regions. For DNA of the same cell, and I forgot to say this is a young cell, for DNA of the same cell, we observe DNA, it's in a more interconnected structure with patches of high DNA. However, these patches of high DNA are less distinctly outlined compared to the patches of RNA. In contrast, in this aged cell, we observe some interesting differences. So we observe a notable increase in the amount of regions that are enriched in RNA. To be exact, there are 14 RNA-rich regions. So most of these regions are spherical, although some of them have depressed centers. Moreover, for the distribution of DNA of the same aged cell, we observed some interesting differences compared to the previous H cell. So now here we see that the DNA is less interconnected. So it's more distinct. It's forming small spherical features, which is a little bit similar to what the RNA is doing. It's just smaller spherical features. So these observations just highlight some of the differences in RNA and DNA distribution between aged and young cells. We also added a new capability to our toolbox using Christel's datasets, which is 3D colocalization visualization. So on this figure, we have DNA is shown in green, RNA is shown in red, and regions where both components colocalize are shown in yellow. As you can see, the yellow colocalization regions are a little bit hard to see. So to overcome this and visualize these yellow regions even better, we used our tool's ability to isolate regions of interest. So we isolated the RNA. So without this isolation, these regions will be a little bit more difficult to visualize. So just to quickly compare, in H cells, we do see a decent amount of colocalization regions. It does vary between cells. So this one we see has a little bit more than this one. But I do want to draw your attention to this particular young cell where there is a lot of regions where RNA and DNA colocalize. Now, this is due to the basically diffused appearance, or not appearance, diffused distribution of the DNA in this particular cell. So we believe that these regions of colocalization correspond to active DNA transcription sites. I want to take a moment to highlight the importance of this tool. So our depth correction tool, along with the new capabilities, have allowed us to explore the 3D distribution of RNA and DNA. So that's something that 2D imaging alone cannot do. So we've also applied this tool to visualize the colocalization between influenza virus, sphingolipids, and cholesterol, because there's been studies that have hypothesized that sphingolipids and cholesterol are crucial during the assembly step of the influenza virus for egress. Now, in the interest of time, I will not be able to go through these results in details, but I will say that using our tools, we were a virus, sphingolipids, and cholesterol, both on the surface and intracellularly. So this visualization capability may help answer other biological questions regarding the roles of these components during viral infection. And the last application, we transitioned to seeing if our tool was applicable to algae cells, which are different from mammalian cells. So the key question that motivated this study was, is there heterogeneity in the incorporations of 15N and 13C labels within individual cells and between cells? So this question is crucial because understanding this variability can inform us about metabolic processes at the single cell level. So 15N, on these experiments, 15N is labeling nitrogen-containing molecules, for example, such as chlorophyll, and 13C is labeling any carbon-containing molecules, for example, lipids, or also potentially chlorophyll. So to basically answer a research question, what we did was that we compared both the levels of incorporation of 15N and 13C within and between cells. And we can clearly see, definitely for the 15N, clearly obvious that there is label or variability in the incorporation of these labels, both within and between cells. For 13C, the same is true, although it is not extremely obvious. Now, we wanted to even look further inside the cell to look at this variability. So then we took cross-sections. So with these cross-sections, we can see how much the incorporation of 15N, it's very abundant on this region, not so much on this region. The same is true for 13C. We see this ring-like structure that's very rich in 13C, but then the rest of the cell is not as enriched. So if you're wondering, what is how do these features look in 3D? What could these features potentially be? Well, we also explored that. To begin addressing this question, we just made these maximum intensity projection images. So basically what these images do is that they highlight the brightest pixel in each XY location in the sample. So by doing this, we were able to at least take a sneak peek and look at how these enriched features look in 3D, but we didn't stop at there, right? We used our tool's ability to isolate 3D regions of interest in order to even better visualize the shape of these features and also get measurements on the size. So say for this particular example, this 15N enriched feature, based on its size, shape, and composition, we believe that it may correspond to the chloroplast. We did the same thing for this particular 13C enriched feature. We can see it here. It's like a little ring-like structure enriched with 13C. So based on shape, composition, and also size, this compartment was located inside what we believe it's a chloroplast. So we believe it may correspond to membranes, maybe the black-white membranes, or if not, carbon-containing molecules such as glucose or CO2. Again, remember, our primary goal is not organelle identification, but rather improving the visualization of these highly enriched features. And I do want to summarize by saying that this tool allows, visualizing the 3D shapes of features of interest. And this can provide information about cellular metabolism or other cellular processes at the single cell level. So to conclude my presentation, I'm very excited to highlight that we developed a depth correction tool that requires no additional data beyond the data that you get when you do nanosims analysis. It's suitable for use with previously acquired nanosims data. It's also time efficient. And ultimately, it improves the 3D visualization of the intracellular features. Now, this tool simplifies analysis by eliminating the need for correlated data and thus improves visualization of 3D features, helping understand the intracellular processes and biomolecular distribution. Additionally, we've introduced new capabilities to our toolbox, which are isolation of 3D regions of interest, 3D visualization, colocalization. So these capabilities greatly improve these tools' utility, enabling more detailed analysis of 3D nanosims images. And I do want to finalize by saying our developed codes for the depth correction, morphology reconstruction are available online for free. You can scan this QR code to access our codes. And with that, I would just like to take a couple of minutes to acknowledge the people who contributed to this work. So my advisor, Professor Mary Kraft, my former lab mate, Rennie Gorman, and Christelle Guillermier. And also, I am extremely thankful to be given the opportunity to present our research work during the seminar. So huge thank you to the organizers of this webinar. I'm very appreciative of having this opportunity to present my work or to present our research work. And with that, I thank you for your attention, and I would be very happy to take any questions.